All right, well, welcome everybody. I wanted to introduce the endomembrane system to you by talking about a group of diseases called lysosomal storage disorders. Individually, these are mostly genetic diseases, and individually, there's there's a really rare chance that you could come down with them. But collectively, there are about 50 documented types, and the odds are about 1 in 8,000 live births. And what makes these diseases so devastating is for a significant majority of them, they involve children and they're not curable, and they will lead to a slow declining death. And by slow, I mean over several months to more likely years. Take Tay-Sachs disease, for instance. Tay-Sachs disease starts to manifest anywhere between 9 months and 2 years, typically. And for those first 9 months of life, you watch your child progress, maybe starting to learn how to crawl, maybe even getting up and walking, and then it starts to degress slowly, where they lose all of those skills, and several years later, finally succumb to the disease. It's a horrible thing to be a part of. To understand why these diseases are so devastating, we need to understand from a cellular level how proteins are made, how they're transported, what they do, and this is going to require a solid understanding of all of the main organelles within the cell. But before we get into that, I want to just highlight the idea of the endomembrane system, why it's so important. When we think of endomembrane, we're talking about membranes or membrane-bound organelles inside the cell. And we need to understand why that's so important. To do so, let's take a glimpse of the membrane. This is an outer membrane. You can see how it's constituted, where we've got an outer leaflet, an inner leaflet, and each leaflet consists of phospholipids. And then we've got a lot of proteins. Shown here in orange are integral proteins, proteins that are anchored inside the membrane itself, or peripheral proteins in orange, the red word, integral. And then, just recognizing each one of these, as we've discussed already, a phospholipid, and embedded within the fatty acid tails that you can see down between those two phospholipids is cholesterol. Of course, phospholipids are amphipathic. That's what allows them to set up this bilayer and act as a semi-permeable membrane, which is very important. There's not a lot of things. Most things trying to go through will simply bounce off. But some things can actually make their way through without any problem. And these are going to be small, nonpolar, non-charged, lipophilic structures. So let's make sure we understand this. So, membrane permeability. What can go through? Things that are small and nonpolar. And recognize, right? Water is a polar molecule. We've learned about water. Water does not easily pass through membranes, so we're gonna make sure we understand water doesn't go through this very easily. Non-charged. So that means ions, things like sodium or potassium or chloride, also on the do not enter list. And this is going to be important in this unit, especially as we set up resting membrane potential, as we set up and watch how action potentials function. So we need to understand those don't easily pass through the membrane. They need help, and we'll talk about that in the next video. The last attribute is lipophilic. Some people say hydrophobic. They're mostly synonymous, but there are subtle differences. Lipophilic simply means I have to love the fat, right? This whole area right here where the fatty acid tails are, in between the layer, if I don't like that, I can't get through. And that's what makes this phospholipid bilayer such a formidable barrier. We can also talk a little bit about membrane fluidity. Because we're on the subject, we need to recognize that fluidity is dictated by two different molecules, the properties of those molecules. Of course, the first being phospholipids. And there are two aspects of phospholipids that we need to consider. Specifically, when we talk about phospholipids, we're talking about the fatty acid tails. Because of intermolecular forces, which we've already discussed, if I increase length, this is going to make it more solid. In other words, fluidity is going to decrease, so that we see that fatty acid tail length is inversely proportional to fluidity. The other thing we have to consider is the amount of saturation. Also inversely proportional, increased saturation leads to a more solid structure, so decreased fluidity. But if I increase the number of carbon-carbon double bonds, I decrease saturation, that will increase fluidity. And then, of course, the other thing that we need to mention, we already mentioned it just previously, was cholesterol. Cholesterol acts as a fluidity buffer by wedging itself in between the fatty acid tails and not letting them get too far apart, but also not letting them get too close together. Cholesterol is an important part of the cell membrane. Now that we have just the overall picture of what the cell membrane is and its key purpose in providing a barrier, let's consider the endomembrane system. So let's pull up a cell. As we look at this image, recognize that the bilayer that we just drew, all of the lines, the yellow outer line of the cell, the white, the orange, the blue, the green, all of these lines represent a lipid bilayer. And because they're all, for the most part, the same in terms of being lipid bilayers, we can squish them together, they can fuse, we can pinch them apart. All of these things, it makes it really, really dynamic. To get back to lysosomal storage disorders, let's start from the end. So down here we have the lysosome. The lysosome, with an internal pH of 5, which is very acidic, compared to the outer pH of, neutral, 7.4. This is an acidic organelle. How does it work? Well, simply because if you remember, charged things can't go through membranes. So if I take protons that are on the outside and I pump them with ATP, I pump them to the inside of this, that decreases the pH. And this is important because in this acidic environment, this is the recycling center of the cell. There's a lot of things that get chewed up. And with lysosomal storage disorders, this is the problem. I'm missing a protein, a functional enzyme of one sort or another that just doesn't find its way to the lysosome, or it gets here and it's mutated and doesn't work properly. And then the recycling center is broken. And if the recycling center is broken, then you can appreciate, right? If your trash isn't picked up during the week, what happens to all the trash in the house? It just accumulates, it gets bigger and bigger. And over time, pretty soon you have an unsanitary situation. It's the same way with the cell. If I can't break things down and recycle them, then the trash increases. And pretty soon that's going to lead to cell death. So over time, sometimes months, sometimes years, cells die off because they're full of trash. And as they die, if it's a neuron, I start losing neurons, and that's going to affect how I walk, how I behave, how I think. It could be cells on the skin. It could be cells in the gut. There's lots and lots of different kinds. But eventually they're going to die, and that is going to lead to illness and eventually death. So in the case of Tay-Sachs we were talking about, the protein that functions as a recycling enzyme, a hydrolase, in the lysosome is called beta-hexosaminidase A. We'll just show it with a beta sign. There's our protein. And its job is to chew stuff up. We've already talked about gluing stuff together. How do you make carbs? How do you make triglycerides? Through dehydration synthesis reactions. These hydrolases, it's just the opposite. Let's add water and split things in half. As we think about this process, the first question that should come to mind is, how did that enzyme, how did beta-hexosaminidase A get to the lysosome in the first place? How was it born? Where did the protein come from? All these questions are what the endomembrane system is all about. So this is our final destination here in the lysosome, but let's go to the beginning now and see if we can't make sense of it. Where do we start? We start in the nucleus. So here's the nucleus. The nucleus does a lot of things, but for now, the most important thing to recognize is that this is where the genetic material that we know as DNA, or deoxyribonucleic acid, this is where the DNA is hanging out. What's the job of the DNA? The DNA is just letters, right? If we think about what this is, it's just A's and G's and T's and C's. Of course, those mean something. Adenosine, guanosine. These are nucleotides, but I only have 4 of them. And it's the sequence of nucleotides that's really the important thing. What is DNA? The DNA is the instruction book for how to make proteins. It tells the cell how to make a protein. And each gene of the 30,000 genes that are in your DNA, each gene gives the instructions for a specific protein. So we're in here, we have our instruction manual, and we're trying to make this specific protein, beta-hexosaminidase A. What we want to do is we want to make a copy of one side of this double strand. We just want to copy one side. There it is. This is now called a messenger, or mRNA. And there's some differences in DNA and RNA. The important thing is RNA is not as stable. It's going to be turned over really quickly, and it's single-stranded. But it is, for the most part, a copy of one of those strands of DNA, where the gene was, and from which I'm going to make this protein. One other thing about the nucleus that we have to consider is the fact that it has actually 2 membranes on it. Remember, all of the lines that we see here, these represent a lipid bilayer. So I have an outer leaflet and an inner leaflet. The nucleus has a second lipid bilayer around it, but the second lipid bilayer is also continuous with the endoplasmic reticulum. And the endoplasmic reticulum can be divided into 2 different types. We have the smooth ER down here, and up above, this one happens to be studded with a little organelle called a ribosome. All these little ribosomes give this a rough texture when looking at it under the electron microscope. And so we call this section the rough ER. So getting back to our story with the mRNA, the first thing we have to do now that we've made that copy, and it is important to know, this process is called transcription. Transcription is copying the DNA to make an RNA. We're going to take that RNA and transfer it out of the nucleus. Here it goes. And we have to find the organelle that's going to now translate the code and turn it into a protein. And that organelle is the ribosome. So that RNA is going to feed right here through the ribosome. Now, what's really happening, maybe if we zoomed in on this, the ribosome itself has 2 parts. It's got a top part and it's got a bottom part. And remember, this mRNA, the best way and the easiest way to think about it is a series of letters. So we've got A and G. Instead of T, because it's RNA, we have to think about U, maybe another U, and then a C, and then maybe another A. As this sequence of letters goes through the ribosome matches, it reads this actually in 3. So it sees this code right here, UCA. It says, oh, I know which amino acid is associated with that code. And so that amino acid comes up into play. And then, as the mRNA continues to pass through the ribosome, we see the other code, right? Here's another one, AGU. Well, that's going to be a different kind of amino acid. So now I have 2 amino acids in play, depending on that 3-letter code, and the ribosome catalyzes the formation of the peptide bond that holds those 2 amino acids together. If you recall from our protein study, this is building the polypeptide chain This is the primary structure of protein, the amino acids all linked together. So now that we see that, I hope you can appreciate this protein itself is being made, this polypeptide chain, right here inside the lumen of the rough ER. That's why the ribosome is docked onto the surface of the rough ER, because this protein is being made inside of it. So now we have this nascent protein that's just hanging out inside the rough ER. This is the beginning of this beta-hexosaminidase A protein. It's, for the most part, still not functional. I've still got to do some things to it. So what's my next step? Well, my next step is to actually leave the rough ER in a transport vesicle. This essentially is just a part of the membrane itself budding off, separating itself from the endoplasmic reticulum, and then, as you can see in our drawing, fusing with this next organelle over here called the Golgi apparatus. Now, we're going to pause just for a second right here and look back at some other functions of these organelles that we need to recognize. First of all, if we continue to remove phospholipids from the endoplasmic reticulum, what's going to happen to the ER over time? I hope you can appreciate that the more I remove, the smaller this structure is going to be. I've got to counterbalance that. So one of the elements of the smooth ER is lipid synthesis. The ER is responsible for making more phospholipids so that as the phospholipid membrane buds off to go to the Golgi, there's always more to take its place. The other thing that's super important for the smooth ER is calcium storage. I'm not going to talk a lot about that now, but we will definitely come back to this as we think about action potentials, as we think about, even more importantly, how muscles work. When we look at the neuromuscular junction, we're going to come back and recognize calcium as a key player in those types of events. The other question we have to answer is, how does that transport vesicle that just budded off of the ER know where to go? How does it get from the rough ER to the Golgi? And that brings up another organelle called the centrosome, or the microtubule organizing complex. The centrosome is an organelle that consists of two centrioles at 90 degrees of each other. And it really has one job for what we care about, and this is to make microtubules. And microtubules, cytoskeletal elements, they're large polymerized structures that act as the highways of the cell. So they grow out like this. And if we actually zoom in and look at this little transport vesicle, it's attached to a little motor protein called kinesin that's got a hold of it and is actually walking along this microtubule. So that's how we know where to take these. That's how it gets to where it needs to be. All right, now let's pick up the story. So we're here at the Golgi apparatus. The protein is now going to go through this stack of pancake-looking structures. What's the Golgi all about? This is all about kind of the UPS warehouse of the cell. This is where processing and shipping are taken care of. There's a lot of proteins going through this process, and the cell has to know where to ship it. Do I exocytosis? Do I send it here? Do I send it there? That's all part of the Golgi. And of course, the processing, we mentioned earlier that this protein coming in probably wasn't folded quite right, maybe needed a couple of things trimmed off of it just to make it functional. That's the processing that goes on in the Golgi. There are post-translational modifications. Maybe I need to attach a sugar, glycosylation, things like that, that will turn this protein into a fully functional protein. So that when it comes out, and it's going to come out through one of these transport vesicles, just like it came in, now it's fully functional. Of course, in this scenario, we're headed down to the lysosome, where this is actually going to fuse with the lysosome, and that protein is going to be placed inside now to do its job. All right, now just to finish the picture, there's other proteins, like maybe, something that's exocytosed. I'm thinking, like insulin or something like that in a beta cell of a pancreas. Let's follow this protein. It's made still here inside the rough ER. We see it in the transport vesicle. We see it go through Golgi, but it's not in this vesicle that's going down to the lysosome. Instead, it's shipped in an exocytic vesicle that's headed out here over to the surface. Or maybe this protein itself is a transmembrane protein. So it's born, and just to keep track of this, we'll put a green dot on the inside. It's born in the membrane itself. It's a transmembrane protein, and it's born right in there. Well, if we follow this protein, right, we can see it. We can see it in there. We can see it going through the Golgi inside this exocytic vesicle. So what's going to happen to these proteins? Well, during exocytosis, these exocytic vesicles fuse with the outer membrane. And what's inside is just going to come out of the cell. There it is. What's in the membrane now becomes part of the extracellular membrane. Maybe this is a receptor that's looking for some sort of signal outside the cell. So this is a very important process. Exocytosis is always happening in order for the cell to function properly. In some cells more than others, certainly. But with the same idea going back to the endoplasmic reticulum, as stuff is leaving, I've got to replace it. Where here, we're adding to this membrane constantly through exocytosis. These vesicles become part of the outer membrane. To prevent the cell from growing too big, what has to happen? Well, I need to reverse the process. So in endocytosis, the cell is constantly bringing stuff in. And we see this endocytic vesicle here. Of course, in most cases of endocytosis, if I'm thinking about pinocytosis in particular, I don't necessarily know what I've just brought into the cell. And I certainly don't want to bring anything that's harmful. So one of the things that I need to do is bring that over and immediately fuse it with the lysosome. Because now it's going to dump all of the contents of what it is into the lysosome to be destroyed. Well, not only am I worried about destroying what's coming in, but the membrane itself, this is how the cell recycles the membrane. Right? I've got new phospholipids going into the membrane, but I'm pulling out old phospholipids to be recycled. That way I don't have old molecules. I'm always fresh. I'm always turning over. And this is where beta-hexosaminidase A comes into play. One of these phospholipid type molecules is a sphingolipid called a ganglioside. And this is what beta-hexosaminidase A is in charge of destroying. Just one type of, essentially, a phospholipid. But because it can't, this starts to accumulate inside the lysosome, and the lysosome gets bigger as it gets fuller and fuller in trash, and eventually the cell dies simply because I didn't have the right protein in there. Now, what happened? Usually, right, when we think about genetic diseases, it's because back here inside the nucleus, one of these regions has a mutation, right? Those four letters that we were talking about, A, G, C, and T, well, maybe instead of a T, there was an A. That message is going to be transcribed on the mRNA, but it's the wrong message, right? It's the wrong letter. Well, that means if we come up here to the ribosome, this is the process of translation. That's going to be messed up because I'm reading an incorrect code, so I'm putting in the wrong amino acids. And if that alters the shape of the protein, it could alter the function of the protein. And indeed, that's what we see. That's why mutations in the DNA cause problems with protein function that lead to genetic diseases. So, in summary, let's just look at the key organelles and think about what they do. The nucleus. This houses the DNA and is the site of transcription, where I'm copying the DNA to make an mRNA. The rough ER, this is the site of protein translation. The ribosome is the organelle that does the translation and is docked onto the surface of the rough ER. The smooth ER, we talked about calcium storage and lipid synthesis. The Golgi, this is kind of like the UPS warehouse, right, where we've got processing and shipping. The lysosome, this is all about recycling. It's got a low pH to do that. The centrosome, the source of microtubules, which are the highways of the cell. Peroxisome, mitochondria, proteasomes, we didn't talk about these in the endomembrane system because they're not part of the endomembrane system. They're not involved in the protein synthesis and production transport that we talked about, but they are important membrane-bound organelles. The peroxisome is all about detox, right? The key word here when we think about peroxisome is oxy, even peroxy, right? This is taking hydrogen peroxide, H2O2, and converting it into water. This is one of the primary means that the cell has to counterbalance oxygen-free radicals that are generated during metabolism. Mitochondria, of course, this is the powerhouse of the cell. So all of the cellular energy in the form of ATP is not all of it, but a significant majority of it is produced here. Proteasome, this is in the cytosol of the cell. Its job is to recycle cytosolic proteins. So it destroys them, breaks them down into individual amino acids that I can then use to make a different protein. Then the cytoskeleton, we mentioned one of those things, microtubules, but we've also got intermediate filaments, like keratins, for example, your skin. The whole reason your skin has the ability to stretch but not break, it's got high tensile strength. This is all about intermediate filaments. And then also the third one is actin microfilaments. So this is all about structure and also motility. The cell being able to get up and move if it needs to. Certain cell types can do that anyways. So there's a list of relevant organelles and a description of the endomembrane system, along with the membrane itself and how it works, to help you hopefully gather a basic understanding of what the cell is all about, protein production and function and the different organelles